Chapter 12, Topic 2.3, the second line of defense. In this topic, I want you to be able to list the four main categories of nonspecific immunity, outline the steps in phagocytosis, outline the steps in inflammation, discuss the mechanism of fever and what it accomplishes, name four types of antimicrobial proteins, and compose one good overview sentence about the purpose and mode of action of the complement system. In this topic, we're going to be discussing phagocytes, inflammation, fever, and antimicrobial substances. Now let's talk about phagocytes and phagocytosis. I'm not going to have you memorize specifically the phases of phagocytosis, but I want you to understand the process and how it acts in innate immunity. Okay, we have a blow up here, we have a little bacterial cell, we have a PAMF, in this case it's the peptidoglycan in the cell wall, and we have on the outside of this macrophage a toll-like receptor that recognizes the PAMF. So it's kind of like when we were talking about mechanisms of pathogenesis. This is like adhesions and ligands, but it's us attaching to and attacking the pathogen or the foreign invaders. So they get stuck to the outside of the phagocyte, they get pulled in and engulfed into a phagosome. So we have them inside of a vesicle. Now we have your lysosomes. Remember the lysosomes from the parts of the cell. They're full of digestive enzymes. They're full of hydrolytic enzymes that you don't want running around damaging the inside of the cell. They fuse with the vesicle that the bacteria is in, and those digestive enzymes digest them. And that's what the cell eats. That's part of how it gets its nutrient. Whatever's left over gets spat out. Now, part of what gets left over is also presented on the surface of the cell. All phagocytes are antigen-presenting cells, or APCs. We're going to talk about that some more later, but this is where they get the antigens to present to other immune cells, is through phagocytosis. So that's what I want you to know. They attach to PAMFs by use of the toll-like receptors. They digest the microbe, they kill them, and they spit out what they don't use and or present what they don't use. Now let's talk about inflammation. Inflammation is part of your innate immune system. It's a response that you get anytime you have injury to the body or the tissue. Now you can get inflammation to physical damage. When you sprain your ankle, you don't have a break in the skin, you don't have a foreign invader, but you have inflammation. The sprained ankle swells up to the size of a grapefruit, and it's part of the repair process. Now, because this is microbiology, we're going to talk about inflammation as it relates to fighting microbial infection. So, just so that you know, that's not the only way you can get inflammation, but we're going to focus on that part. Okay, so I'm fixing chicken for dinner, and I've got raw chicken sitting out on my plastic cutting board, and I cut myself. And so there's a few salmonella, little bacteria, and who knows what else on the knife, and it's injected into my skin. Okay, so I've got damage to my skin, I've got damage to the dermis, I've got damage to the blood vessels. So what happens is, first thing, a clot forms, stops the leakage of blood, heals the breach temporarily. Okay, so an abscess starts to form. We also get vasodilation and increased permeability of blood vessels. When the damaged cells release cytokines such as histamine, prostaglandins, etc., but they're released anytime you have damage to any of these cells. This stimulates your capillaries to dilate. The cells separate so that more lymph leaves, more fluid leaves, and you start to get swelling. Okay, this allows monocytes that have been floating around to wiggle through the cells in the capillary, get in where all of those lovely salmonella bacteria are, and begin to phagocytize them. Now the first responders, because there's more of them in your blood, are neutrophils. They're going to get there first, and they're going to start phagocytizing first, and they release more cytokines to say, hey, there's a problem here, come here. So monocytes come in, they mature into macrophages, and they start phagocytizing. Now, as things develop, okay, we're going to say that our macrophages and our neutrophils have taken care of the problem. They've eaten all of the bacteria in the area. Other cells come in and repair my cut. I get a blood clot, i.e. a scab up here, and depending upon how well I've cut myself, scar forms. Okay, and things return to normal, with the exception that I now have more macrophages hanging out here waiting for more trouble. Because they're thinking, if Teresa's dumb enough to cut her finger here, she's probably dumb enough to cut her finger here again. Okay? So, the signs of inflammation, okay, are generally related to the increased flow of fluid to the area of damage. Okay, you've got swelling, that makes sense. You've got more fluid going there, that area is going to swell. You have redness. Okay, that's because you have increased permeability of your blood vessels. You've got more blood flowing into the area. More lymph. You have heat. That's also from the increased fluid from further inside of your system. You're bringing warmer fluid to an area that's generally colder. And then you also have pain. This fluid buildup has a tendency to press on nerve endings. Okay. Now, we're going to talk more about the damage caused by chronic inflammation. But here we're going to end with acute inflammation where the hazard has been averted, we've taken care of the invaders, cytokines are not being released anymore, and everything resets and goes back to normal. Now let's talk about fever. Fever is often associated with inflammation. Sometimes it's not, but let's talk about why your body would raise its temperature. That's what a fever is. Okay, this is going back to the chapter on bacterial growth requirements. Bacteria prefer a certain temperature. Most of your cells prefer a certain temperature. All pathogens are mesophils. Okay, and if they're pathogens of humans, they prefer 37 degrees C. By raising the body temperature, you're bringing the temperature above their optimum temperature. It's generally still within their cardinal temperature, because if you raise the temperature to the point where you're denaturing bacterial proteins, you're also denaturing the proteins of you, specifically heat-sensitive cells like, I don't know, your brain. Okay, that's why it's good to keep a temperature within 39 degrees C. If you get to higher temperatures, then you start having problems with your body, you start having damage to the brain, you can go into convulsions. That's why we keep an eye on fever, because you don't want it to go too high. So, you raise the body temperature, you raise the temperature above the optimum temperature of your pathogen, and it slows down their growth, their division. This also causes higher metabolism of your immune cells. They actually prefer 39 degrees Celsius. So your neutrophils and your macrophages actually phagocytize and function better at these higher temperatures. Okay, so once you've had the problem taken care of, you want to be able to lower your temperature back down to normal once the hazard has been averted. That's why a long-term low-grade fever is not good. If you have a low temperature, a low fever for short periods of time, that's actually good. You don't want to mess with that. But if the fever gets into higher areas, higher temperatures, where you're going to cause problems to the patient, to you, then we use drugs to lower that. Okay, so that's fever, and that's how it functions in innate immunity. Now we're going to talk about antimicrobial substances that function in this second line of defense. We're going to talk about complement, interferons, iron-binding proteins that humans produce, and antimicrobial peptides. Let's talk about complement. This figure actually has more information that I want you to know. I mainly want you to know what the goals of complement are and how they're caused. Okay, so There are three pathways for starting the complement pathway. There's the classical, which involves antibodies. We're going to talk about that more in the next chapter. There's the alternative, which we're going to talk about here, and then there's the lectin. Lectin is a molecule that is produced by macrophages in response to certain paths. So it's basically the macrophages saying, hey, there's a problem here. So let's mainly talk about the alternative pathway. So complement are various proteins that are floating around in your blood that complement the innate immune system. So they float around, and when they're stimulated, then they kick in and they help the innate immune system. So if they're stimulated, parts of these proteins are going to cleave. I don't expect you to remember that C3 cleaves into C3b and C3a, and they go and do different things. But we'll just say that part of these proteins go and they stick to the surface of your pathogen, of your invader, and this enhances phagocytosis. We call this opsonization. This comes from the term for tasty. So it's kind of like sprinkling salt on your food. It makes it more tasty and you eat it faster. So that's kind of what's happening. But what's actually happening is it makes for little handholds for the macrophages to grab a hold. They have receptors for complement proteins. So if they see complement proteins, they're able to grab a hold and phagocytize better. Now, some of the complement binds to your basophils, to your mast cells. This causes a release of histamine. This causes inflammation, which recruits your neutrophils and your macrophages, which causes more phagocytosis. Now, in later chapters, we're going to talk about histamine and how it functions in allergies and why we take antihistamines to reduce inflammation. But right here, this is how inflammation is stimulated, okay, by complement. Okay, now others cleave, they form proteins, and they form a channel, okay, in the cytoplasm of your invading pathogen. We call this a membrane attack complex, or a MAC. That's one of the functions of complement, is they form these pores, these channels, which causes outflow of the cytoplasm of the cell. It causes cytolysis. It causes lysing of the cell. Okay, so to review, what I want you to remember, what stimulates the complement system is there's the classical pathway, which is stimulated by antibodies. There's the alternative pathway when complement recognizes a PAMF, okay, and kicks in, and then there's the lectin pathway. Now, the results of the complement pathway being activated are opsonization, inflammation, and MACs. Next up are interferons. Interferons are very cool, and we're using them highly to treat chronic viral infections. They've also been used in treating cancer. Let's talk about how they do it. Okay. Interferons are naturally produced by your cells. What happens is I get a virus, it comes in and it takes over the viral, the cellular processes to make viral proteins. Okay. Well, when this starts to happen, the cell knows that something's not right, and it produces signals that go to the neighboring cell and say, hey, I'm done for, I'm dead, I've been invaded, but you can still save yourselves. So the neighboring cell, while this cell has been converted to making more viruses, this cell starts making antiviral proteins, or AVPs, okay, in response to interferons that are produced by the infected cell. So when the new viruses come and infect the neighboring cell, these antiviral proteins bind to ribosomes, preventing any protein synthesis. This keeps the virus from making its own viral proteins. Eventually it dies. It gives up in about 2 to 3 days, and then the neighboring cell can go back to its normal functioning. Now, you feel horrible in this process, but it prevents those invading viruses from making more of themselves, and it limits spread of the virus. Now, this is useful in treating cancer because we make interferons in the lab. We inject them into a cancer patient or a patient that has a chronic infection like hepatitis B or hepatitis C. And this shuts down any protein synthesis that is going at a rapid pace. Cancer cells are growing rapidly even when they're not supposed to, so if we shut down their protein synthesis, they can't cut back and they die. That's the plan anyway. So that's interferons. Now let's talk about antimicrobial substances, those iron-binding proteins that carry iron from place to place. Now you remember from the previous chapters, cytophores are proteins that pathogens make to bind iron. Well, we make iron-binding proteins, and there's kind of a tug-of-war between the pathogen and us. But I do want you to know our iron-binding proteins. Transferon is a protein that carries iron around the blood. Lactoferrin carries iron within milk, tears, any secretions. Okay, so a mother can transfer iron to her growing baby. Ferritin binds to iron in your bone marrow and in your spleen, any place where red blood cells are being produced, or like in the spleen, you have worn out blood cells are degraded there. The iron is recovered and it's transported to the bone marrow where you're making new blood cells. And of course, hemoglobin binds iron in red blood cells and transport. It is used in the transport of oxygen within your body. Alright, now to talk about antimicrobial substances, specifically antimicrobial peptides. Now, their function is to inhibit cell wall synthesis. This is mainly effective against gram-positive bacteria. They have that thick peptidoglycan cell wall. They also produce pores in the plasma membrane, so in that way they're similar to complement when it forms membrane attack complexes, or MACs, and they also destroy DNA and RNA. That prevents replication or division of your pathogens. Well, that's it for this topic. Remember, I want you to be able to list the 4 main categories of nonspecific immunity, outline the steps in phagocytosis, outline the steps in inflammation, discuss the mechanism of fever and what it accomplishes, name 4 types of antimicrobial proteins, and compose one good overview sentence about the purpose and mode of action of the complement system.